Hello, welcome to this session all about ARSACS. But first, I would like to turn it over to Sonia Gobet from the ARSACS Foundation to give us a welcome on behalf of their foundation.
Thank you, Celeste. Hello, everyone joining in this morning. My name is Sonia Gobet. I am the co-founder of the Ataxia of Charlevoix-Saguenay Foundation, Fondation de l'Ataxie Charlevoix-Saguenay. I'm based in Montreal, so I co-founded this foundation back in 2006 with my husband, Jean Groslo. And the mission of the foundation, which is based in Montreal, is to finance research projects to eventually have a treatment for the people affected with ARSACS. Actually, we are now in a call for proposal for all the researchers out there listening this morning. Please send in your application. We're looking for people to join this big international project. So back to you, Celeste. Thank you.
Thank you, Sonia. On behalf of the National Ataxia Foundation, welcome to the session all about ARSACS. It is our absolute pleasure to partner with the ARSACS Foundation to provide these two sessions all about this disorder during the month of February. But before I introduce our two wonderful speakers we have for you today, we have some housekeeping items to go over first. So if you wouldn't mind, Dr. Dupré, going to the next slide, thank you. First, if you need closed captions in order to better interact with or understand the session, you can turn that on at the bottom of your screen. And please feel free to use the chat to interact with different members of the community if you have any questions or would like to connect with them. But if you have questions for either of our speakers today, we ask that you put that in the Q&A window, which you can also find at the bottom of your screen. Sometimes questions can get lost in the chat, and putting them in the Q&A window will save them all in one place for our Q&A session at the end of this talk. So thank you in advance for helping us out on the back end, streamline things a bit. And with that, it is my pleasure to introduce our two speakers today. So first is Dr. Nicolas Dupré, who is a neurologist at the Québec-Laval University, who is a full professor in the Department of Medicine at the School of Medicine at Laval, and the director of the Neuromuscular and Neurogenetic Disease Clinic of the CHU de Québec, Université Laval. Dr. Dupré completed his medical training at McGill University and his residency in neurology at Laval University. He completed his postdoctoral training at Montreal Neurological Hospital and Institute and at Harvard. In 2007, he discovered a gene, SYN1, implicated in a new form of hereditary ataxia and has since been highly recognized internationally. Dr. Dupré is now involved in several provincial, national, and international networks that contribute to the discovery of genes involved in neurological diseases. He has generated over 100 original publications. Dr. Dupré's remarkable achievements have earned him several awards, including the 2015 Quebec Society of Muscular Dystrophy Award, ALS component related to his involvement in the clinic for neuromuscular and neurogenetic diseases. So he is our first speaker. And our second speaker is Dr. Élise Duchesne, who is a full professor in the physical therapy program at the School of Rehabilitation Sciences, Faculty of Medicine at Université Laval. She is also a researcher at the Center for Interdisciplinary Research in Rehabilitation and Social Integration, the Québec Université Laval Research Center, and the Interdisciplinary Research Group on Neuromuscular Diseases. With a background in muscular physiology, and physical therapy, Dr. Duchesne focuses her research on rare diseases with muscular impairments, particularly on using translational approaches to bridge the gap between laboratory findings and clinical application. Her work is primarily centered on rare neuromuscular diseases such as ARSACS, which is why she'll be speaking with us today, as well as oculopharyngeal muscular dystrophy and myonic dystrophy type 1. She is involved in several groundbreaking studies, including a multi-center randomized controlled trial investigating the effectiveness of rehabilitation intervention on rare diseases. And with that, Dr. Dupré, Dr. Duchesne, the floor is yours.
So good morning, everyone, and thank you for the National Ataxia Foundation to allow us to present on ARSACS, which is a common recessive ataxia, particularly in northeastern Canada and the province of Quebec. So ataxia is usually referred to as a lack of order in movement. So it's important to consider that this lack of order in movement can affect the eye control. It can also affect the attention span people have on specific tasks, cognitive tasks. And it also affects the limb movements, upper limbs, lower limbs, And so it's important not to consider this concept as a restricted concept to cerebellar diseases. So it's a very wide array of functions that are affected, and that's why research is so important in that field. And many disorders cause ataxia. So there are different ways to classify ataxias, and one of which we proposed, which is after we did a systematic review on autosomal recessive ataxias, because this field has grown very widely over the last few decades. We went from recognizing and knowing only a few of recessive ataxias to now having a whole tree of ataxias that share very particular features. Some of them have mainly cerebellar effects. Some of them have intellectual disability. Some of them have mitochondrial and metabolic syndromes. Some of them have extrapyramidal symptoms. Some of them have peripheral nerve function affected, and also motor neuron function. So a whole array of peripheral and central nervous system impact in these diseases, and so it makes them very complex diseases to study and also eventually to find therapy for. So ataxias in Canada have always been part of the research spectrum, somewhat caused by the founders of this research field in Quebec, whose name is André Barbeau. He described some of the rare ataxias in Quebec that we have today, but mainly because there are founder effects in the population of French-Canadian origin, which allowed initially to make these studies more rewarding, because the gene finding was not as fine-tuned as it is today. And so there are a number of ataxias that we find in the French-Canadian population, but now some of them have been described in Quebec, and after that, usually they have been described around the world thereafter. So there have been studies to try to find the frequency of ataxias, and surprisingly, we don't know that much about the frequency of ataxias in each individual country. Usually, it's a complex epidemiologic study that you have to do. You have to combine your many different clinical scales, and you have to combine data for rare diseases. So we have a clearer picture in Eastern Quebec, but this is a long way to go before we get a whole worldwide pictures of how frequent ataxias are in each area. So this is the province of Quebec, for those who are familiar with North American geography. So Quebec City is in this area. So this is one ataxia on which we worked several years ago that we presented last year at the NAF Foundation. And it's just to put ARSACS in a whole context of ataxias. And there are so many research groups working in the field of recessive ataxias that eventually, if you find a path that leads to a therapy in one disease, it may lead you to the path to a therapy in another disease as well. So this is just a recent review we did in one of the Nature review in Neurologist to show how complex this tree of recessive ataxia has become, and just to show the part of ARSACS in a very wide spectrum of diseases. And this is for those who are interested in biology and function, just to show you how one specific disease category, which is a rare disease category, can give us answers about the function of cellular networks on a very wide way. And this goes from the mitochondria to the nerve cell. And this is only showing one type of cell, but there are multiple types of cells involved in ataxias, and especially in ARSACS, which affects the central and the peripheral nervous system. So there wouldn't be a story of ARSACS if there hadn't been this meteorite that arrived 300 million years ago in the Charlevoix area. It created a flat area where people were able to establish, you cannot colonize a mountain range. So if you want to establish yourself somewhere, you need a flat land. And this is how these people arrived from France. And one of them is a descendant of these founders, Dr. Jean-Pierre Bouchard, and he's the one who described ARSACS some time ago in 1978. So we go back a while, with the history of ARSACS, and it was one of the first well-characterized recessive ataxias worldwide. So, there's quite a few decades of knowledge and research that have been accumulated through times. And this is just to show you where usually French Canadians come from in French, usually from northwestern France. So ARSACS is characterized usually by early onset, but there have been There are frequent cases which still I can diagnose in our population in their advanced 20s and 30s. So it's the disease that you still should be looking for in adults. It has characterized by slow eye movements with speech problem with that we call dysarthria. It's often characterized as well with by brisk reflexes, so pyramidal involvement, and there is an axonal sensory motor neuropathy, and that's important because in recessive ataxias, you may have an effect of the peripheral nerve like you have in ARSACS, but you may not, like you have in sign 1 ataxia. So it's one of the characteristic differentiating features. And we have known the gene for more than 20 years. For now, it's the SAX gene, and so it's readily available in most gene panels. So how do we detail this disease? So it's a slowly progressive cerebellar ataxias, which usually will start, it can be noted early on in life, after a few years of life, and the gait unsteadiness for the most severe cases can be noted early on. Spasticity in the lower limbs will usually come a little bit later, and then you have the peripheral neuropathy. And what you will notice is some distal wasting and weakness of the hands and of the feet that will be characteristic of the disease after maybe less than 1 decade. And when you do an MRI on these patients, they have an atrophy of the vermis, which is the upper portion of the cerebellum, and this is very characteristic. So if you have an atrophy of the vermis and you diagnose a peripheral neuropathy on EMG, you really have to think of ARSACS. And in areas where ARSACS is very common in Quebec, clinicians will know about it. But if you are in an area where ARSACS has never been shown or diagnosed, and there's still many of these areas around, it's one disease to look for if you have these clinical characteristics. And often when we work up some young children or young adults with degenerative disease, we send them to an ophthalmologist. And if the ophthalmologist describes retinal hypermyelinated fibers, then it's quite pathognomonic of ARSACS. So now you can do optical coherence tomography as well. Sometimes this technique is available to ophthalmologists, and it has also some characteristics that can lead to a diagnosis of ARSACS. So this is just a table that shows how diverse the disease is and how severe the effect can be on many, many systems. You have cognitive decline in a portion, you have effects on speech, on swallowing, you have some epilepsy in some cases, spasticity, the neuropathy, the weakness, the pes cavus, the scoliosis. So it's really a disease which has multiple effects. That's why we need to find a therapy maybe for more than one of these effects, especially if you're talking about symptomatic therapy. If we had a disease-modifying therapy, the goal would be to have an impact on all these effects at the same time. And usually the patients, when they are followed by clinicians, will be part of different spectrum of disease. Some have a very, very prominent cerebellar impact. Some have very prominent neuropathic features, and some more prominent pyramidal features. So it's never exclusive, but some patients can really vary in the clinical presentation. It's important to be aware of this. And these are the findings that will be described by ophthalmologists. And when we do MRI of patients with ARSACS, there are different characteristics that we can find. And the MRI in ARSACS can be differentiated from other ataxias. But I would say that radiologists that are very accustomed to seeing degenerative diseases may point to some cues that are specific to some diseases, but often you will have to rely on your knowledge as a neurologist to really pinpoint the aspects. And in ARSACS, you have some pontine hypointense aspects that you can describe in patients. You have also the vermis atrophy. So that's a differentiating feature from other ataxias, but still the radiologists that are not accustomed to these diseases will probably not be able to point out the specific disease. And this is if you do a spine MRI to a patient with ARSACS who has been evolving for many years, you will see that the spinal cord is atrophied, and you can see how devastating this impact can be on the long run on the function of patients. when we look at population genetics, obviously this gene, so we could say that we are now at the 25th anniversary of the discovery of the gene of ARSACS. It's always very important to discover the gene of a disease, but it's a long way before often you can find therapies, going from the disease description in 1978 to identifying the gene in 2000. So that's almost 25 years. And to eventually find a therapy, we're 25 years after the discovery of the gene, and we don't have a gene therapy yet. So it's a long time. It's a very cumbersome process. Even if we have founder effects that I described, in this meteorite area in Charlevoix, there is a lot of diversity of mutations. So when people talk about targeted gene therapy, sometimes gene therapy may function for one specific mutation. So imagine the number of mutations that have been found in ARSACS, and this is not this is an outdated article. This is to show the mutations in the French-Canadian population. They're already very diverse, even if it's a founder population. And these are other mutations that have been described worldwide. So it's a long way before we find a targeted gene therapy for ARSACS, because often you have to find a treatment for each specific mutation. And so we may have to look for other means or other pathways before that. So the clinical presentation and early evolution has been described, and there's a wide array of presentation. That's why you should not overlook this diagnosis, even if patients are in their 20s or 30s. Sometimes they have been misdiagnosed with other types of diseases, and so you have to keep on looking for them and reassessing your genetic profiles. So this disease has been found in many populations, so it's quite common in Holland. So for those who are joining us who are from Europe, there are populations where it's been more frequent than others. These are the typical eye findings that you find in patients. And so in that area of Europe, it's quite common. There have been cases also described in Brazil, and they also find similar features, the eye findings. So we are starting to see really this disease becoming more and more global. And this is a description of the very typical vermian atrophy and the pontine signals that are altered. So the clinical features are very characteristic, and these are mutations that have been described in other countries. And if we extend and we start to do exome, usually we used to test genes one by one, and then we ordered panels for ataxias, and now people are starting to, for rare diseases in their population, to use exome or genome sequencing. And when you do these approaches in a more systematic way, eventually you can regroup, and see where the disease has been described worldwide. And you can see that it's really described among all the continents. And so it's going to be, this community is going to certainly increase its size over the years. Even a case that's been described in India. So we are really adding up the worldwide aspect of the disease, and that's why communities have to come together. And I believe these seminars are very, very important to do it. So the pathophysiology and disease models, for those of you who are not biologists or scientists, it may be more difficult. So I will try to adapt the level. But this again is the atrophy of the vermis and the pontine hyperintense signals. So this is how we used to assess patients on MRIs with, and we still do it today. But there are some better imaging techniques that are coming out, and this is from the ProSPACS study. And it's important for the patient community to know that there are studies out there that try to evaluate in a more specific way the features. So the goal is it's not necessarily to aid in the diagnosis, but it's eventually to find markers that could be used for clinical trials and to see how the disease affects the brain and its whole impact. And so it's important to participate and get involved in these diseases eventually. So there are studies also in mouse models that look at Purkinje cell vulnerability depending on where the cells are located. And this is just to show some mice studies that are done. And all the multiple pathways that have been involved. So we see SAXin there, which is the gene causing ARSACS, and it's involved in the regulation. You know, we see HSPs, we see exosomes, we see tyrosine kinases, we see riboproteins. So you know, you may want to target SAXin itself or a specific mutation to cure the disease. But you may go in different pathways like this and try to affect the downstream events of the mutation. It may actually be easier, and you may have an impact on more tissues, and you may not have to go gene by gene. So there are really different approaches that have to be considered, and it's not, gene therapy will not necessarily be the whole answer. Because SAXin has an impact on different systems, it's important to recognize, is it more the mitochondria that's involved, or is it more processes of autophagy? So it's very important to understand the biology of the disease so that we can use the tools we know of biology to try to correct the downstream effects of these mutations. So for those who are not scientists, again, you can see how complex. You try to link, you know, the SAX protein to many other gene functions. Sometimes you may want to alter the function of one of these genes around, and then eventually it will help to mitigate the effects of the SAX mutations. So the turnover in the protein is important. So you may want to affect the turnover of the protein in a very early effect using all particles that affect mRNA degradation or mRNA stability. You may want to have an impact on the lysosome, on the microtubules. And also, the mitochondria is important in most neurodegenerative diseases, and it's not necessarily the one factor that will have the most impact, but it's one factor that you have to mitigate. So I'm coming back to the HSP, and I won't go in very. So I spoke, you know, about studying different models of ARSACS. There is the model of there are mice models, but there are also models of zebrafish. You know, people look at these small zebrafish and try to rescue SAXin mutations to see if they will alter the function of the zebrafish in a positive way. So I think it's important to consider all these models together. And in terms of getting ready for trials, you know, eventually you want to show an impact on a mouse model, usually of one of the molecules that you're trying to test in human. And there have been some studies looking at mitoQ, for example, the mitochondrial function. I would say none of these studies are definitive. They're rather small studies, but this is the way. Usually, animal studies will lead the way before we try anything into human trials. And there are other aspects that you want to look at, calcium homeostasis. And eventually, people probably have heard of this technique that was described some years ago of using pluripotent stem cells. So the Purkinje cell is a so the Purkinje cell is the one that has the most one of the most important function in the cerebellum. And most diseases that affect the cerebellum, the ataxias have an impact that's relevant to Purkinje cells. So you want eventually to develop Purkinje cells model that are in vitro. So using pluripotent stem cells, for those who are not familiar with it, is you will take a cell from a patient, usually one of its blood cells, but there are other types of cells that can be used, and you will revert it back to a pluripotent stem cell that eventually you will redifferentiate into a human cell, but in vitro. So it's very important to develop these cellular models. They're very expensive, but eventually you will have an in vitro model on which you can test molecules. So it's nice to have an animal model, but still these are, you know, cells from less complex brains. If you are using Purkinje cells that are derived from human cells, then you can put them in vitro and start to build what people have called mini brains, where you put cells together and eventually study the brain in a very systematic way and an in vitro way that reproduces the human brain in vitro. And this is a review paper that we just published this December 2024. For those who are interested, that looks at the pathophysiology of ARSACS and looks at how we can lead the way eventually with iPS-derived models. So this is to show you again how you go from patients to the dish, Petri dish, pluripotent cells to the Purkinje cell, which is in the cerebellum, to eventually building models that reproduce the brain, and eventually to look at therapies and therapeutic developments. And you want to go back to the patient, obviously, eventually. And these are other ways to diagnose the diseases. Somewhat that may, when you get back results from genetic tests, sometimes these results have not been validated and biologically, so you get a result which is indeterminate. So you can use other tests to validate the mutations, and that usually will allow you to have a diagnosis which is more reflective of the biology of the disease. And so this way, each mutation can be validated, and you can even use these tests to make diagnoses of patients. This is another group that looked at the similar way of diagnosing patients. So it's always interesting, to go from the patient to the therapies. Right now, we are usually, we do not have a disease-modifying therapy. So you need, obviously, to be evaluated by a neurologist eventually, who will confirm the diagnosis, do different tests like an MRI or an EMG. You will need occupational therapy and physiotherapy to help you with mobility, development, assessment, help with speech therapy for the dysarthria, evaluation for hearing. And so the follow-up is very important on these patients in multidisciplinary clinics. Obviously, if in your environment ARSACS is not well known, a general ataxia clinic will be fine. And so eventually your clinician, if he's not familiar with the disease, can contact the patient from our area. And sometimes we can give clues as to how to confirm the diagnosis or to initiate some therapies. There are other medications that you can give for spasticity. And so it's important to treat these patients in a multidisciplinary context. And these are studies that try to look at rehabilitation programs that my colleague, Dr. Duchesne, will speak about later. Some scale that people use to evaluate patients, usually for clinical trials. And some more, I would say, fancy ways to look at the important clues that we have from patients, that we can evaluate. And eventually, if you have more data, you may be able to really find the important aspects that will make a clinical trial successful. These are the groups, the Saguenay group, which are very active in studying diseases because obviously that disease is more common in that area. And some of the international collaborations that look at ARSACS globally, like the PROSPAC study and Treat ARCA. So these can be found on the ARSACS Foundation website. And this is just to emphasize what Mrs. Gobey spoke about earlier about the ARSACS Foundation. It's a worldwide ARSACS Foundation, and it also has a registry now on which patients can enter some minimal data sets, and this allows the ARSACS Foundation to really try to link up the community affected with ARSACS worldwide. So obviously, together, we can make a difference. I will now allow my colleague to complete her talk.
Yes, thank you, Dr. Dupré. I want to to thank also the National Ataxia Foundation and the La Fondation Charlevoix-Saguenay for giving me this opportunity to present about therapeutic approaches in ARSACS. So Dr. Dupré gave you a very nice introduction of the disease. So from now, it's easy for me to present you what we need to target in a physical therapy approach. So as Dr. Dupré mentioned, so there's a lot of impairments that characterize the disease. So cerebellar, pyramidal, and neuropathic systems are impaired. So that lead to significant motor impairment. So as Dr. Dupré mentioned, there's a lot of other systems that are impaired, but from a physical therapist perspective, those limitations are very important to treat. And importantly, those significant motor impairments lead to difficulty performing activities of daily living and social roles or of patients having ARSACS. So it's very important to take care of that impairments. Unfortunately, there is no curative treatment to slow down the progression of the disease, so rehabilitation is a key alternative by being low cost and also accessible. So next. Thank you. So a few groups around the world have published systematic reviews in various types of ataxia, and those review aim to determine what is the effect of rehabilitation programs on physical impairments. So those systematic review reveal improvements in limb coordination, physical function, balance, and functional mobility in various types of ataxia. So that's very encouraging for our ARSACS population. So we published a pilot study in 2022 specific to ARSACS. So we trained patients for 8 weeks, and we also show improvements in balance, trunk control, disease severity, and activities of daily living. However, only few studies are specific to ARSACS, and the consequence of that is the lack of all quality studies to develop recommendations and guidelines for clinical management. So our team recently developed 2 training programs, and those were developed based on the natural history study result that we obtained. So the GRIM, the Groupe de Recherche Interdisciplinaire sur les Maladies Neuromusculaires, received funds from Fondation de l'Ataxie de Charlevoix-Saguenay to understand what is the natural history study of ARSACS. And based on those results, so based on the magnitude of the muscle impairments that were assessed, we developed rehabilitation programs that target specifically those impairments. So the first program is called PACE ARSACS, and this program is a home-based program, and the other one is the IMPACT program, which is a clinical-based rehabilitation program. So I will present you those 2 programs today. So the first one, PACE ARSACS, the home-based one. So it's this program has also been funded by the Fondation de l'Ataxie Charlevoix-Saguenay. So this program is a home-based exercise program specifically adapted for this population. And this program is quite suboptimal because when we created this program, we had 2 main considerations in our head. The first one is no risk, and the second one is no fall. So this program relies on an initial standardized assessment by a physical therapist, and based on this assessment, a decision tree is used to assign a rehabilitation program with the appropriate difficulty level according to each participant ability. So this program is individualized and is carried out at home independently. So this program consists of 3 domains that are trained. So sitting balance, standing balance, and transfer. For each domain, there is a number of levels of difficulty that are included in each domain. And the program is carried out 3 times a week for 12 weeks. And follow-up by a phone call was made every 2 weeks to ensure security and progression. So here I just want to show you some characteristics of the population that was included in that study. So 14 participants were included, and an equal number of men and women, and also an equal number of walker and non-walker. And I want to point out the adherence rate. So you can see that there is a lot of the adherence rate is pretty high. So reaching 98.6% for the non-walker and almost 99% for the walker subgroups. So this program consists of two phases. The first one is a control phase, and the second one is the intervention phase. So all participants were assessed before the beginning of the control phase, after the beginning of the control phase, and thereafter they carry out the intervention, and obviously all participants were assessed at the end of the intervention. During the control phase, patients were asked to maintain normal daily physical activity levels and continue their usual routine. And during the intervention, the people carry out the exercise program at home for 3 sessions per week. So I don't have time to present you the results of the control phase, but I can say you that no changes occurred during that 12-week period, and it's not surprising because it's a slowly progressive disease. So we expected to measure no changes. And during the intervention phase, one of the main outcome is that no falls occur. So it was a very we were happy to see that result. So if we present the result as a group, so here you can see the result of the whole group. So all the participants are included in that table. So we assess balance by using the Berg Balance Scale, the Ottawa Sitting Scale, the ABC Scale. We assess mobility by measuring 10-meter walking test at self-selected speed and maximal speed. And also the 30-second sit to stand. And we also assess participation with the life age and the ataxia severity with the SARA scale. So you can see here that we only measure significant result for participation and ataxia severity in the sense of the hypothesis, meaning that we measure an increase in participation and a decrease in ataxia severity following the training program. For mobility, we measure a significant effect, which was in the opposite of the hypothesis. I mean that people walk slower following the training program. And it's not the first time that we saw this kind of result in our study, because during 12 weeks, people are trained to have a clearer movement. We ask them to do movement slower, but better. So we were not very surprised to see that in a 10-meter walking speed. So as Dr. Dupré mentioned, so ataxia, ARSAC is a very multisystemic disease, and there's a lot of heterogeneity in that population. So it's very hard with 14 participants to see significant effect of a training program. So we decide to analyze the result differently at individual level. And we found that very cool paper published in 2021 that suggests an analysis tool to capture a broader assessment of clinical benefit in heterogeneous complex rare disease, such as ARSACs. So the principle is quite simple. So for each domain that is measured, so for each outcome measure, for each participant, you determine a threshold. So, for example, the domain 1 for the threshold for domain 1 is 54 meters. So in our study, we calculated the standard error of the measurement, which is an indicator of a true effect that appears clinically. And we look at each measurement for each patient if we were outside the standard error of the measurement. And if yes, we calculated the total score of outcome that were outside this threshold. You can go next. Okay, thank you. So here are the individual results. So if I explain you the first the first box. So for the Berg Balance Scale, so the BBS for the patient number 1, which is a walker. So we measure an improvement of 3 points out of this scale after the training program. And this improvement is outside the standard and error of the measurement. So we color this box in green. So all the green boxes are the results that were improved following the training program. The yellow box were stable, and the red box were worsened following the training program. So we divided the participant into walker and non-walker. And if you go next, I will just summarize here. So our results show that 60% of the participants improve following the training program. And out of those, out of the patient that improve, 5 out of 6 non-walkers improve, 3 out of 8 walker improve, and only 1 out of 14 participants did not improve in at least one outcome measure. And there is another way to interpret that table is to look at the outcome measure. So it's very useful to look at this when we are planning to determine outcome measure to be included in clinical trials. So if you see here in the total whole cohort, so 5 out of 8 outcome measure captured changes. And those effects are more pronounced in non-walkers. If you go next, yeah, thank you. So I will give you the example of the Ottawa Sitting Scale. So you can see in the whole cohort that 43% of the whole cohort improve in that outcome measure. If you look for only for the walker, you will see that this number is not that high, so only 12.5%. But if you see in the non-walker group, you will see that 83% of the participants improve. So there is difference between walker and non-walker, and this is exactly the same thing for the life age, and the same thing for the SARA. So it's important to consider the characteristic of the participant to see if they, on which outcome measure they will show improvement. So we were also interested by patient voice. So we asked at the end of the training program how they find this training program. So I just give here some quotes that we receive, but in general, the program was very well accepted by the participant. So the take-home message from this program is that rehabilitation is beneficial for individuals with ARSACS. The home base seems suboptimal for walkers, so the most improvement were seen in the non-walker participant. And we were not very surprised by this because this program focused on safety. So of course, we didn't expose walker participant at too much risk. So that's the hypothesis why non-walker improve more. So as I mentioned, it was highly acceptable for all participants. And if you go next, so you can see that this program is available online. So we have 2 versions, so French, English, and another language is coming. So we made this program available online to be sure that every clinician around the world can use it if needed. So I will just show you some slides from the other program, which is a clinic-based rehabilitation program. So it's an ongoing randomized controlled trial. So we have the result right now for 38 participants out of the 68 participants that are targeted. So this program was a supervised program, so the stimulus was optimal. So each participant is with PT during the training session. So once again, the initial standardized assessment is done by a physiotherapist, and the rehabilitation program was in a swimming pool and a therapy room. So 2 times a week, people went into a rehabilitation in a therapy room, and 1 time a week in a swimming pool. So once again, 3 main domains were trained, so postural control, balance, and functional mobility. And we developed a standardized exercise inventory allowing progression throughout the week. So the dosage is the same than the other program, so 3 times a week for 12 weeks, and the evaluators were blinded. It's okay. So once again, I show you here the attendance. So the attendance was 97%, and we were trained participants that I will show you the preliminary result that are either walker or non-walker. No, sorry, that are all walker. Sorry. So once again, it's the same. We did the same individual analysis at this stage, and this cohort improved. 60% of the cohort improved. 9 of 16 participants improved, and once again, only one participant did not improve in at least one outcome. So for the outcome measure, we saw that 4 out of 8 outcome measure capture changes. And this time we can see that the balance, so Berg balance scale, Ottawa sitting scale, and ABC scale improve, were outcome measure that improved the most. And in this study, ataxia severity were most stable. So once again, we were interested by the acceptability of this program for the participant. And I just put here some quotes. And once again, this program was highly acceptable by patients. So the take-home message for this clinical-based program is, once again, that rehabilitation is beneficial for individual with ARSACS. A supervised rehabilitation program seems more optimal to challenge walking capacity, seems acceptable for all participants, and of course, more result analysis to come, including sub-analysis based on walking capacity. So in conclusion, pilot data suggests differential positive effects of home and supervised program for walker and non-walker individual. We found important to develop those two programs because it's not all participants that have access to supervised physical therapy, including a pool. So that's why it was important for us to develop a home version of training program. Interventional studies can help identify appropriate outcome measure for clinical trial. Interventional studies can help in planning patient stratification for clinical trial. And as seen in other diseases, rehabilitation could be useful also in combination to pharmacological approaches to come. So thanks, everyone. And I also want to thank my team. So back to you, Celeste.
Hey, Dr. Duchesne, thank you so much for a fantastic presentation. We are going to jump right into questions, but as a reminder, if you do have a question for either of our speakers, please put it in the Q&A window at the bottom of your screen. So our first question, someone asks, they have been diagnosed with ARSACS, they have heard this is a genetic disorder. Should they have their kids tested for ARSACS?
So the meaning is so the patient has ARSACS, right? This is what I understand. Obviously, if you have ARSACS, your children will be carriers of the disease gene, of one allele of the disease gene. So this means they will be like your parents. So this is, you know, just a fact genetically. So what will be important is when they decide themselves to have children, they should have their spouse tested to know if they carry an ARSACS mutation. So if they don't carry an ARSACS mutation, this is done kind of routinely in areas of high prevalence of ARSACS, like in the Saguenay area. So if the spouse does not carry any of the known gene mutations, so the likelihood of having a child with ARSACS is close to 0. If the spouse does carry, like let's say you marry someone to which you are remotely related, this will increase the odds as well. Then the risk will be 25%. So your children will be carriers. They will not have the disease, but they have to be eventually. Their spouse has to be tested to know if they are carriers, but the likelihood of having a spouse carrier, if you're not living in an area where ARSACS is common, is very, very low. So if you have ARSACS, the likelihood of having a child or a grandchild affected is very low, but the risk can be assessed specifically by a professional geneticist who will go through that with your children.
Thank you for explaining that. We have a question from another attendee. What are the differences between someone whose symptoms start in childhood versus adulthood? we have someone who was diagnosed in his early 30s, and he's wondering why it took so long for the symptoms
Yes.
to start.
Yes, it's very, very interesting. When I showed this diagram, you know, where you have the three circles, you know, if you have a severe neuropathy, you're likely, this means effect of your peripheral nerves with your feet being deformed, and you're probably going to have a more severe disease. If you don't have a severe neuropathy and you have mostly cerebellar symptoms and mild spasticity, it may take more time. So, I would say that it's severity, but also the type of phenotype, and this is really a struggle. You know, your remaining gene pool will want to fight off this this missing function of ARSACs. So, depending on your genetic background, you may have very different impact of the disease within the same population with the same mutation, but also within the same family. Sometimes you may have later onset and earlier onset. So there's still a lot of unknowns.
On the topic of genes and different mutations in them, we had a few people ask, since there's multiple mutations that cause ARSACs, if they've already been diagnosed, they've had genetic testing, should gene tests be done multiple times to confirm or check to see if they have more mutations?
So you will, if you have a confirmed disease mutation, that's it. You know, it's like a letter written on, you know, and a very, very, so it will not, it's not useful to test it again. Sometimes people have a mutation which is likely. And in that case, you know, let's say you have been diagnosed 20 years ago with a mutation which was not well validated. Sometimes it's a good idea to test again to make sure that but only if the mutation was not known or not validated. So I've had patients, for example, where the diagnosis was not clear, and then we tested again, and we found a mutation which is pathogenic. So the tests evolved, but the mutation will not change over time.
Thank you for letting us know. We have a question for the two of you. People are interested on how they can get involved in ARSACS research, or if there's options to participate virtually in the studies that either of you are conducting, if they live far away. Can you talk a little bit more about how they can find out more information on ARSACS research and the opportunities for participation in your own research initiatives?
Yes, I can go where I can answer that question. So there is a site that is called clinicaltrial.gov. So this site gathers all the studies, the ongoing studies, and on this site you can see the clinical trial and also the clinical studies, which is not pharmacological studies. So you can see all those initiatives there. So sometimes there is a limit regarding the geographical area where you are living, and it's supposed to be all written in the site. And if not, you have contact of the PI of that study, so you can contact the PI to verify if you can be enrolled in that study or not.
I would say another element could be to log on the ARSACS Foundation website and to just give your name as, oh, I have ARSACS and I'm in this area. So if the ARSACS Foundation website, let's say, identifies 100 patients in a given U.S. state, they will wonder, you know, what's going on. You know, there's a lot of ARSACS in that area, so it may give scientists an incentive to contact even collaborators in that area and initiate a clinical trial site. So wherever you are around the world, you know, you log on and you put your name out there to say, you know, I have ARSACS, and it's very simple, very easy way to put the community in touch, but also eventually to identify communities where the prevalence is higher.
We are also tracking that information here at NAF. So if you have already registered with us, if we get any information about ARSAC specific studies, we will email that to you. We also have a step-by-step guide of how to navigate clinicaltrials.gov, because it can be a little bit confusing the first time or first 3 or 4 times you use it, I'll ask our staff member to put a link to that guide in the chat. We have another question around the PACE study. Could you please clarify what non-walker in the PACE study means? Is this a non-walking person or a person who is using different mobility devices or has never used a walker?
Yeah. So we define walker and non-walker based on the ability to walk using or not walking aid. So if a patient use a cane or a walker, they are considered as walker because they can walk even if it's not independently indoor. So the only threshold is using a wheelchair, for example, that will fall into the non-walker participant.
And then we have another person who asks, shares a little bit first. Our current physiotherapist does not believe in doing PT just for the sake of doing PT and wants to write PT for a specific goal. This seems contrary to the results of what you're sharing. Do you recommend ongoing or continual PT for ARSACS patients, particularly young ARSACS patients?
Yeah, I see. And the one problem in the academic field, so we can do things that is not 100% that we cannot apply into the healthcare system due to, for example, limitation of resources and everything. And I know that in healthcare system, the PT are most used for the evaluation and be sure that people are well assessed for changing a walking aid, for example, or going into wheelchair. So I know that the focus is more oriented toward this. And one thing that is, I think, obvious is that it's very hard for PT to be convinced to give a home-based training program because this population falls a lot. So it's very it's frightening to say, okay, do exercise at home, and so you have a responsibility that, okay, I say this to the patient, but the patient fall a lot. So that's why we want to develop a program that we know that was suboptimal, but we want to be sure that no fall occurred due to the program. So I say that, yes, I believe a lot in exercise and doing everything. And for the young ARSACS participant, so you can use the sense of play with a little one to be sure that they are in movement and that they improve by movement.
And one final question for Dr. Dupré. We've had a number of questions from people asking about yearly MRI scans. Is this something that they should be doing or asking their neurologist for, or if there's any other kinds of yearly checkups they should be doing with their neurologist if they have ARSACS?
So a yearly MRI would be a big waste of time and money, so don't do that. So the MRI is useful when you're looking initially at trying to diagnose a rare disease, and that will pinpoint areas where, you may suspect a specific type of ataxia. But once you've done it initially for the workup, it's not to be done other than when you participate in a specific trial like the ProSPAC trial that does yearly MRIs to try to better understand the structure. But these are not this, they're not the standard MRIs. They're MRIs that are research-based, so the analysis is much more detailed. Otherwise, I think the multidisciplinary clinic is important, the targeted impact of a physiatrist at a given time, of a speech therapist at a given time, and all the people that surround either in pediatric or adult. I think that's what's important. And eventually, when clinical trials come, and if the clinical status of the patient is less severe, then the odds of going into a trial will be higher. So everything that maintains function, independence, and also when you're young, keep on studying, do what you have to do, listen to your parents, future is for you, and when you're older, you want to keep your independence, and so it's important to structure the care so that when we reach therapies, patients are as in shape as can be,
Well, that is all the time we have for today. Dr. Dupré, Dr. Duchesne, Madame Gobet, thank you so much for joining us today and giving us this talk on ARSACS. We really appreciate your time and your expertise. And to all of you who spent the last hour with us, who learned a little bit more about ARSACS, I hope you found something valuable from this session. And wherever you are in the world, good morning, good afternoon, good evening. Have a wonderful rest of your day. Take care, folks.